Our next speaker is Emma Canaday, A Song of Fire and Water, evaluating the dispersal potential of the cave-obligate pseudoscorpion Hesperochernes mirabilis. I love that title.
Thanks. Let me get my screen share set up. Is everybody able to see everything and hear me?
Yes.
Okay. As I was introduced, I'm Emma Canaday, and I will be presenting my research that I did during my undergraduate at Southern Adventist University, looking at the dispersal potential of a cave-obligate species of pseudoscorpions. So some background on pseudoscorpions. These are small predatory arachnids, and they have been well documented to use this behavior called foracy, where they will attach themselves to a larger, more motile animal and use their movement as a dispersal vector. This even allows for potentially long distance dispersal, where these pseudoscorpions might be moving into areas that aren't inhabitable to them, but are able to move from cave to cave, say, into habitable regions. This is really interesting considering that recent genetic mapping, which hasn't been published yet, has shown that there's connections between caves as far away as northern Alabama and Indiana. So for a species that has never been identified outside of caves, they're traveling an incredibly long distance. We proposed two possible methods for this dispersal. The first is foracy on bats. We think that they might be able to attach and then move with the bat during their summer migration. We also think that dispersal through water could potentially explain submovement, as many cave systems are connected by aquifers, and a pseudoscorpion could be washed through these. All of our specimens were collected from Oaks Cave in Tennessee, and we established, first off, a control condition, which was cave-like conditions. Then, to test the potential for foracy, we wanted to see if they could actually survive summer conditions that a bat might be moving through. So we set up a trial that had heat and light conditions similar to the summer of Alabama, the hottest range that these bats would be going through. And then to check the ability of pseudoscorpions to move through aquifers, we tested how long they could survive completely submerged in water. So that was similar to our cave conditions with complete dark and a cold environment, but they were completely submerged in cave water. Now, the control and the thermal groups, we were able to check survivability by prodding them with a paintbrush daily, and if they responded at all, we took that as a sign that they were still alive. Unfortunately, the water group didn't show any signs of life while they were submerged, but if you took them out and dried them, they would resume responsive behavior. So instead of checking daily, we removed 10 samples every day and checked their survivability and subsampled until we reached the end of our living specimens. This concluded our experiment 1, but because the water group was non-responsive, we couldn't feed them the same way that we were feeding the control and the heat survival groups. So we introduced our experiment 2 to reduce the variables between our trials. After experiment 1 was done, we took all of our remaining specimens from our heat and control trials, re-randomized them, and reran the experiment, this time without feeding them every 7 days as we had the first time. And this way, they more closely resembled our water group, which couldn't be fed during the experiment. After all of our trials were completed, we sent our samples to Auburn University to Charles Stevens, who identified the life stage, sex, and body size of all of our specimens. Looking at the beginning of these results, we can see our groups of life stage. So we have our control adults and nymphs, our heat adults and nymphs, and our submersion adults and nymphs. We see that the submersion groups are dropping off the fastest as these lines go down, their chances of survival are decreasing over time measured in days. So our submersion group is dying off the fastest, and our heat group is surviving surprisingly well, especially for the adults, as they're very similar to the control. And that was a very surprising result considering these are cave-dwelling specimens that are used to living in a very thermal and light controlled environment without fluctuations. We also saw this in our Weibull model as our heat group survived slightly shorter than our control, but still better than our submersion groups. We also see that the nymphs appear to be surviving better than the adults in submersion, and that might tell us that different life stages have a potential for dispersal via different ways. For the adults, after they reached adulthood, we could also identify sex, and we reanalyzed the data looking at sex between groups. So we see here that this time the water groups showed a high similarity of data. The males and females responded to water as well, very similarly, but the heat groups showed a surprising result in that the females were surviving much better than the males as time went on. And we can see that the males are dropping off while the females are staying very close to the control. Our Weibull model confirms this again, showing that submersion was significantly worse than control and that heat is approaching significance. We see that here that the control males were surviving almost significantly better. Our results were really approaching significance for the heat versus control, but the females were surviving much better, and that we see a strong overlap between control and heat here, showing that the females are able to survive this incredible thermal stress very well. And then again, we see that the submersion group is surviving several days, but not nearly as long as our heat trials. Moving into experiment 2, which introduced starvation to reduce the number of variables between trials, we see that we have our 4 groups here, which were randomized from experiment 1. So we have specimens that were placed in control after being in the heat for experiment 1 versus control after being in control, and on and on. We see a strong overlap between all of these groups, and starvation doesn't seem to be adding a very strong factor to the survivability of these species. They seem to be able to survive similarly, regardless of whether they have been given a food item or not. We see that again as all of our groups have non-significant p-values. And we see that even the shortest period of survival, which was around 15 days, was still much longer than our survival in water, showing that starvation is probably not accounting for that increased mortality in water, that the stress of being drowned alone was probably the cause of that increase in mortality, not the stress of not receiving a food item. Moving into our discussion and really assessing whether these dispersal methods are a viable way to explain that long distance dispersal that we saw, moving all the way from Alabama to Indiana. First, we're looking at submersion. Our median survival for adults was about 4 days, and starvation does not account for the difference between our control and the water. Starvation, the stress of the water alone was probably the cause of that increased mortality. We also saw that nymphs were surviving slightly better than adults, with 1 nymph still alive on day 15 after being submerged completely for 15 days. Overall, the dispersal potential for water seems to show that they might be able to move short distances or within closely connected cave networks, but probably not that long multi-state trip that we're seeing through genetic mapping. Moving into the heat trial, we have some exciting results here. Our median survival was 27 days, which again is just really astounding considering that these are specimens that are living their lives in a thermally controlled, completely dark environment, that they can survive this incredible heat and light stress for such a long period. We see that starvation does not decrease that median survival by too long. So even if, say, they were attaching to bats and they weren't able to hunt during migration, they could still survive an incredibly long time outside of caves. Finally, we saw that females might be surviving slightly better than males. And this is really interesting considering, as we see on this gravid female pictured here, they will carry their egg sac with them until maturity. So a female that was using 4SE to move to a new cave system might be able to introduce all of her offspring with her, and thus that longer survival period might be selected for. Finally, we introduced some GIS modeling to really get an idea of what this dispersal potential means for how far they could move. So each of these black dots represents a cave that has been confirmed to have Hesperochernes mirabilis in the literature, and all of these outgoing circles and shapes represent the max migration of a different bat species. All of these migration periods are about 2 to 9 days at the maximum, so well within the survival time that Hesperochernes could survive outside of a cave. So here we see Moyatis saudalis, which is the Indiana bat. We have Moyatis lucifugus, which is the little brown bat, and Perimyotis subflavus, which is the tricolor bat. So all of these, we can see that they might be able to move from these northern Alabama, northern Georgia region caves up into Indiana and reach these caves that we see gene flow with between these northern Alabama caves. Also, potentially, we've seen a lot of papers looking at caves in the southeastern United States to identify Hesperochernes. But if we look at this potential dispersal map with a map produced by Clover and others in 1999, we see that their potential range is much larger than where people have been looking for them so far. And as they move into farther and farther caves, they could potentially move with bats and increase their range even further. So we see that this species, even though believed to be cave restricted, has a potential to move enormous distances across the United States. Moving into future research, because this was an undergraduate project, we were limited on time and resources, and there are some areas that I would love to see looked at further. So the first thing that we would add to this is a longer treatment period. Potentially over time, we might see a larger separation between males and females as they progress with increasing stress. I would also love to test this with gravid females, as the stress might be affecting their egg sac that they're carrying, and with more nymphs to see how heat was affecting stress. Stress was affecting life stage. We would also love to add by combing bats for pseudoscorpions. If we were able to identify pseudoscorpions moving on bats in the US, they've been identified in other countries, but not in the States yet. That would add a lot of natural history data to our experimental work. Also, potentially, if we could look at the predictive powers of our maps and see where bats are migrating through and identify caves where bats migrate versus caves where bats are not known to migrate, and look at the populations of pseudoscorpions between those caves, we could get another really important piece of natural history data to add to our experimental work. In closing, I'd like to thank Dr. Nelson, my advisor at Southern, for helping me through this research, Charles Stevens, a pseudoscorpion expert at Auburn University, for sharing his expertise on this species, Dr. Corbett and Dr. Carvalho for helping us with the more complicated aspects of the statistics that come with survival analysis, and Chris Ogle of the Tennessee Wildlife Research Agency for helping us get into these caves to collect our specimens. I have my citations, and I believe I still have some time for questions.
Yes, thank you, Emma. That was so interesting and well timed. Let's see. Which one is the first question? Here we are. Excellent work, Emma. Question: how do you see declining bat populations affecting pseudoscorpion vagrancy and population fluctuations?
So that was something really interesting that we discussed a bit. As the bat populations are shifting because of diseases like white nose syndrome, they could be affecting a lot of other populations that rely on bats. So not even just pseudoscorpions, which might have a limited range because of bats, but also all of the animals that live in the bat guano. All of the specimens that we collected were found in bat guano. So as those guano piles decrease, all of the animals that rely on that habitat could also be affected. The bat declines could affect huge cascades of wildlife, not just the bat species themselves.
Okay, if the second experiment was done with individuals that survived the first trial, do you think that could bias the result, as it is only considering the already more resilient individuals that made it to the end of the heat trials?
There is a potential there, but also, as we saw, something that helps me still have confidence in this, that starvation wasn't affecting the water trials, is that the median survival in the water was 4 days, while we were feeding specimens every 7 days. So they were dying off before they would have hit the first feeding event anyways. But the starvation trial does still help us know that even if we have slightly selected for the more resilient individuals, even within those, they're able to survive significant periods without a food item.
Is it also possible one widespread population could have retreated into many dispersed caves?
So one wide, I'm trying to, I want to make sure that I'm getting this question right. You're asking that they could have originally been a larger unified population that then was separated into caves more recently. One idea for that, one limitation for that would be that these populations have a very recent genetic connection. So that work was done by Charles Stevens for his PhD. The connection between here is much more recent than a geological event creating caves or an evolutionary event where they're becoming cave specified. This is a much more recent gene connection than any geological shift. So it seems like they must be moving. It's too fast for a different method.
Great, thank you, Emma.